Well, good morning, everybody, and thanks for inviting me to give it this keynote lecture. It's a great pleasure. So I'd like to start immediately why we should study the rhizosphere. Well, the rhizosphere is actually the most active portion of soil. So it's one of the most important scientific frontiers that we should address to actually overcome the challenges of climate change and population growth. So there's a really urgent need to understand the processes going on in the rhizosphere. So as I said, it's the most active portion of soil because it's actually the volume of soil which is influenced by the root activity. So it's a very dynamic, very peculiar soil volume, which is both in time and in space. So it changes continuously. In fact, why does it change continuously? Because it's continuously shaped by mainly three actors: the plant, the microorganisms, and obviously the soil. And all of these actors, they change continuously and they create these intimate interactions, biochemical, physical, and also chemical interactions. So very, very dynamic. So the importance of knowing rhizosphere processes, so why should it be important, obviously? So for instance, if plants experience nutrient deficiencies, they might show an unbalanced uptake and a different distribution of other nutrients, as it is shown here in the figure. So knowing rhizosphere processes, one can also modify nutrient availability. And that's why I wanted to show this is quite a famous picture, I would say, quite old, still from Volker Römhild back in the 80s. But this clearly shows you how you can influence the rhizosphere processes by this example, for instance, just by changing or by changing the nitrogen form of the fertilizers you apply, you might have as a consequence a different pH of the rhizosphere. So by applying ammonium, you might acidify the rhizosphere, or by applying nitrate, you alkalize the rhizosphere. And obviously by changing the pH of the rhizosphere, this has a huge impact on nutrient availability, since nutrient solubility is a function of pH. So that's a huge effect you can actually have. But obviously in the rhizosphere, what else is there? There are root exudates. Root exudates are low, high molecular weight organic compounds released by the roots. They are all kind of different compounds, and they have different functions, but they are also involved in nutrient availability. They mobilize the nutrients. They are involved also in the uptake mechanisms. But these root exudates, to measure those root exudates is quite a challenging task, especially in soil-grown plants. Why is this so challenging? But first of all, they are readily degraded by microorganisms. They are adsorbed onto the soil particles. So finding an appropriate method to measure and to determine these exudates is quite a challenging task, especially in soil-grown plants, as I just said. In fact, just recently we developed, let's say, a new method to sample the root exudates from soil-grown plants and to measure them. So this is a DGT based method, and it gives us the opportunity to sample the whole root system, but also single root zones. So actually you can choose whatever you want to sample, and it gives you the opportunity to have a kind of map. So it is also an imaging technique. So the result of this technique is that you can actually cut this gel into pieces, analyze it, and this method what we develop is actually based on phosphytrate. So it's a specific phosphytrate, and as you can see, this method then can generate these nice maps around the roots. So basically what you can see is that the root exudation is definitely changing along the root axis, and it's depending also on the root type. So obviously now the next steps will be to develop new DGT-based methods for other organic acids, or also for other root exudate compounds or exudate classes. Obviously that will be the next steps then of our research. But these root exudates, like here we say flavonoids, organic acids, but they're huge, a plethora of organic molecules. They are released by the plants. So this is quite a cost-intensive thing for the plants. So many researchers, well, let's say few, not so many actually, they hypothesize, and some of them demonstrated also, that the plant possibly reacquires those exudates. So getting a feedback, so some energy, let's say, back for the plant. So we wanted to prove this as well. So what we did is we used carbon-13 labeled root exudates. So this experiment, we carried it out with tomato plants. We grew tomato plants both in phosphorus deficient condition and in a full nutrient condition, that's plus P condition. And we supplied those tomato plants with 13 carbon labeled root exudates. We chose different compound classes. This slide shows you just the results of glycine and amino acid, but we tested also organic acid like citric acid and malic acid. What you can clearly see here, that indeed when the plant is resupplied with glycine, it reacquires it. And you can see here that the delta 13C increases. For those who are not familiar with isotope ratios, that means the more positive the delta C 13C is, the more the heavier isotope is acquired by the plant. In fact, by looking at the mass balance, and you compare the same conditions, plus P minus P, at the same concentration of glycine we supply to the plant, you see almost 30–40% increase in delta C13 acquired by the plant. So it's also depending on the nutrient status of the plant, and it's also depending on the concentration of the root exudate, how much it is reacquired by the plant. But are root exudates actually efficient also in mobilizing nutrient? That's not obviously then the next question, because once they are released, are they then actually efficient in mobilizing them? Well, actually then we performed this experiment. It was again a Rhizobox experiment with white lupin. And on this, let's say, different root zones, we applied again a DGT gel, which mimics the root uptake, assessing the bioavailable fraction. Once we deployed the gel onto the root, after 24 hours, we removed this gel and we analyzed it by laser ablation ICP-MS. So by doing this analysis, we got a kind of imaging of the elements which have been mobilized by the root exudates around the roots. So what are the results? Well, here, the first image you can see here in the slide is the hotspots. Let's say I call it hotspots because those colored dots are the regions where we found a higher amount or higher content of iron. Next element is copper. You see again those colored spots indicating that there we have a higher concentration of copper. And this is exactly around the roots. You can see it here. It's most likely that the soil has covered slightly the cluster root of this white lupin. And you see the distribution of the colored spots is slightly different depending on the element. But the interesting thing is what we found is for manganese. And you see here this huge, it's not really small hotspots, but it's a huge, let's say, block. It's like hotspot, a huge hotspot. So we know that actually this white lupin exudes like citric acid, malic acid. So the exudates are the same, but the mobilizing efficiency actually depends on the type of element. And this gradient generated by this mobilization effect is completely different depending on the element. But obviously in the rhizosphere, we also have microorganisms. So we cannot exclude to consider also microorganisms. So we started also here quite simple experiments because considering also only one microorganism. So we inoculated plants with one bacteria. We chose for these experiments a plant growth promoting rhizobacteria, namely Rhizospirillum brasiliense. Of course, in the rhizosphere, we have a myriad billions, thousands of bacteria, but obviously for the experimental process, you need to start in a simple way and then complicate it step by step. And And for these experiments, we performed with cucumber plants, and cucumber plants, again, in full nutrient conditions and in iron deficient conditions. Now, cucumber is a dicot plant, so it takes up iron in a reduced form. So iron needs to be reduced at the root surface by iron reductase. So what we looked at is the physiological alteration induced by the inoculum of this bacteria. Well, as you clearly can see here, take a look first on the left side on this green box. You see here in iron sufficient condition, also the PGPR induces an enhanced activity of this iron reductase enzyme. But in iron deficient condition, this enhancement is much more evident, especially if you see here in iron deficient condition, if these plants are also inoculated with the PGPR. And you see also the area, so the intensity of this activity is much greater, but also here, obviously, you see only one rootlet, but we analyzed the whole root system. Indeed, what we observed is we found an increased iron reductase activity, and this both in terms of number of spots per plant, but also in total spots area per plant. So also this intensity, so the ability of the plant to reduce iron. Dicots, especially in iron deficient condition, they also acidify the rhizosphere, so they release protons. So again, what the Rhizospirillum brasiliensis did is to enhance this acidification activity also in iron sufficient condition. Obviously, again, as for the iron reductase, also in for the proton release, it was extremely enhanced in iron deficient condition when the PGPR was present. And again, both in terms of number of spots per plant and in total spots area per plant. So the Rhizospirillum had actually a really impact on the physiology of the plant. But what does it mean? Does it actually, the plant also take up more iron? So what we did is we supplied those plants with labeled iron, so with a 57 iron, which is a stable isotope, and we measured the uptake of this iron. Well, what we could see is that this Rhizospirillum brasiliensis did not only affect the physiology, but it extremely increased also the uptake of iron, and this fourfold, both in iron sufficient and in iron deficient condition. So it really had an effect. But obviously we were all, still talking only of one deficiency, so of one only nutrient. But obviously in the field, especially in the field, plants face not only one deficiency, but very often combined deficiency. But what happens actually in combined deficiency conditions? So we approached this aspect with two plants, barley and tomato, again, a monocot and a dicot, in 4 different conditions. Control condition means full nutrient conditions, minus P, minus phosphorus, minus iron, and a combined minus P, minus iron condition. So obviously, due to time restrictions, I only show you some results, obviously, of this study. So looking at the analysis of the shoots of the elemental composition, we did this principal component analysis, and I mean, we expected this. So the 2 plant species were separated, but interestingly is that the tomato clustered in 4 different, let's say, again, clusters. So every single nutrient status has its own behavior. On the other hand, barley showed only 2 clusters, and they kind of also overlapped. So we had 1 cluster of plants, the iron deficient, and the combined deficient plants, and control and minus P plants clustered together. And also, what was interesting, who was responsible for those for this clustering? If you look at the first component, we had obviously phosphorus, which was in some cases missing, but also some other macronutrients, magnesium, calcium, and sulfur, while on the second component, we had zinc, copper, and manganese, which are those who are interacting with the iron uptake indeed. So we'll look also on the root exudates. Again, these are the results of tomato plants. So we analyzed the organic acids. First of all, here you see succinic acid. Well, succinic acid was not actually related to any nutrient stress. It was released throughout the experimental period, but was not related to any nutritional stress. Interesting, the other two organic acids, namely malic acid and citric acids, they were related only to -P in the combined deficiency. And that was quite interesting. So in iron deficient condition, we did not observe any malic and any citric acid. The fourth organic acid we detected in the root exudate was oxalic acid. Oxalic acid turned out to be rather constant, raised rather constantly during the experimental period, except for the last sampling point. Indeed, it was highest in the combined deficiency, lowest in the control. So it seemed also to be released at a different time scale. So also the time is definitely important for or affecting the release of root exudates. The last compound class we analyzed is the total phenols. And again, it was enhanced in all the nutrient stresses, but it was not related to a specific nutrient stress. And it was, in fact, lowest in the control plants. But obviously, what happens if the nutrient reaches toxic concentration? I mean, that happens also very often in agricultural soils. And here I want to show you a case study, namely copper. I mean, copper is a problem in many agricultural soils, especially in apple orchards and in vineyards, due to the application of copper-containing fungicides. And this is then especially a problem of when we change the cultivation, since young grapevine plants, for instance, vineyard renewal, the young grapevine plants, they have a shallower root system, so they get in contact with the copper in the upper soil layer. Or it is obviously a problem also if we change the cultivation. So what we did is we choose different rootstocks, and we looked at the effect the copper has on its rootstock, but we were also interested on looking at the possible agronomic practice that could alleviate this copper toxicity. And based on preliminary results and based also on literature data, we've thought and we hypothesized that intercropping with grasses like oat could be a valuable strategy. So as you can see here, these are the results of the Ferkal rootstock, which was one of the rootstocks we tested. And you can clearly see with increasing copper concentration, the biomass clearly was decreased. And you see also a kind of copper toxicity on the leaves. Well, if these rootstocks are intercropped with oat, you still see a kind of a decrease of this biomass, but the plants look much healthier. They have a greener color of the leaves, so the toxicity is not that severe of the copper. Indeed, if you looked at the compartmentalization of this copper, as actually also widely described in literature, the main target of copper are the roots. So both the symplast and the root apoplast. But the interesting thing is, because we were interested in was the intercropping actually effective in reducing this copper accumulation? And indeed, we found that intercropping significantly decreased the copper accumulation in both the root symplast and the root apoplast. So it was effective. But obviously, we wanted to look also to the other nutrients, because maybe copper toxicity affected also the uptake, the accumulation of other nutrients. And what we found out was also quite interesting, because we found a rootstock dependent unbalanced root uptake. Indeed, for the Ferkal, in both conditions, the mono and the intercropped condition, we found an antagonism with manganese and zinc, and an synergism with iron. On the other hand, the other rootstock we tested was this 96.17. We still found an antagonism with manganese, but for zinc, we didn't find an antagonism anymore, but a synergism. So again, these extremely genotype dependent. So when we talk about copper toxicity, we definitely need to consider the type of genotype. So the last example I would like to show you is this quite large study we started last year. It concerns apple orchards, again, copper accumulation. So we decided to sample different sites of apple orchards in our region in South Tyrol, which was a very intensive, where we have a very intensive apple production. And we have also quite high copper concentration in these soils. So we decided these 21 sites, which had also a kind of copper gradient, ranging from a couple of milligrams per kilograms up to 600-700 milligrams per kilogram. So what we were interested in the effect of this different copper concentration on soil quality parameters, especially soil enzymes. We are also interested on the soil microbiome, but I still don't have the data because we got the sequencing data just a couple of weeks ago, so we're still working on them. But I wanted to show you the results of the soil enzyme activities, and this was also quite interesting. As you can see here, these bunch of enzymes, namely exocytinase, exoglucanase, beta glucosidase, and the aryl sulfatase, I hope you can still read it, they are all correlate together, and they are also correlated to the nitrogen content and the soil organic matter. But they did not depend on the copper concentration. On the other hand, the protease and the phosphatase, first of all, they were negatively correlated, but they correlated also with copper concentration. So for the protease, it was a positive correlation. With the phosphatase, it was a negative correlation. This was quite also interesting. And then I'm really curious now what happens next, also especially to the soil microbiome. So I already coming to my concluding remarks, I hope still in time. So obviously the importance of knowing the rhizosphere processes, well, why is it important to know the rhizosphere process? Obviously to predict the nutrient dynamics, and not only the cycling, but the availability, and ultimately also the uptake and the allocation of the plants. And this is due to an increasing world population, which we obviously need to feed. But we need to feed those people by producing on soils, on agricultural land, but this agricultural land is unfortunately facing a lot of soil degradation, and it's obviously not increasing. So on the contrary, we are facing more and more soil degradation. So thus we actually need plants with a higher capacity to accumulate nutrients. We need plants with a higher capacity to reach adequate yields under also adverse environmental condition, and with a higher nutrient use efficiency. So that's why we really need to deepen our knowledge on how plants acquire and use the nutrients. And that's why we need to deepen our knowledge, especially concerning rhizosphere dynamics, rhizosphere processes, because it's actually at the soil-root interface where all the important processes are going on, which ultimately affect our nutrient uptake, and then also our the quality and the yield of our agricultural processes. And if we really understand and know these processes, we are also able to exploit these processes by manipulating the rhizosphere to manage and enhance the acquisition of the nutrients by higher plants. So with these statements, I'm actually at the end of my presentation. Before I thank you for your attention, I also thank all the group here in Bolzano, which obviously helped in getting all these data, or also our collaboration all around Europe, all around the world. And obviously, I thank you for your attention.